This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 46, recorded on November 29th, 2012. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. This Week in Microbiology is sponsored by ASM, the American Society for Microbiology, the world's largest membership society for microbiologists. Find out how ASM membership can help advance your science, your career, and your network. Go to asm.org/advance. Joining me today from Small Things Considered, Elio Schaechter.
Hi there. How are you, everybody?
Well, we're all dried out finally here.
You know, about time. Are you really... Is your house on in New Jersey emerged from the devastation? Or?
Well, it's intact, but the electrical system is all messed up from the salt water because it was flooded, and we have to... that's going to take a lot of work. So it's going to be dark all winter, probably.
Oh, boy.
But it's okay. Some people lost their homes entirely, so it's all right. Also joining us today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent and Elio. How are you all today?
You're back home, aren't you?
I am back home after
For a change.
For a change, after two exciting meetings and a visit to my collaborators in Santiago.
Nice.
Where we're doing a study at a children's hospital at Roberto del Rio and asking some really fundamental questions about whether or not copper will actually help control hospital-acquired infections in a pediatric setting. So we're really excited about starting that trial, and they're really excited about moving forward. And so it was a really interesting trip, and it's always nice to connect with colleagues. And, you know, when you travel away, you do hardcore science for a week, your head wants to explode, but at the end you have loads of fun and just doing the work. And I think, you know, for those of us in science, we're fortunate in that when you have an opportunity to collaborate and you have to get the stuff done, it's really a lot of work, but it's also very gratifying to see it come together very quickly.
That's why we do it. Also joining us today, I have a guest who is a colleague of mine here at Columbia University in my department. Well, it's not mine, but it's the department I'm in. I don't want it, actually. It's too much of a headache. The Department of Microbiology and Immunology. Jonathan Dworkin. Welcome to TWiM, Jonathan.
Thank you, Vincent. Elio, Michael, it's nice to meet you guys.
I've known you for a long time, but this is your first time on TWiM, of course. I've heard you talk about your work, and that's what we're going to do today.
All right.
Are you a New Yorker?
I spent most of my formative years in New York. I was not born here, but I sort of grew up here, yes.
Which borough did you grow up in?
In Manhattan.
And you went to college here? Or
I did not go to college. I went to Swarthmore College in Pennsylvania.
Swarthmore?
Nice. Oh, boy.
Isn't that where David Baltimore went?
It is where David Baltimore went, yes.
So did you
know...
I didn't overlap
Bob Savage? Did you know Bob Savage there?
He was there just... yes, I knew him when I was there, yes. He's a great guy.
Old friend of a terrific guy, yeah.
And you were a physics major at Swarthmore?
I was a physics major. Yeah, I actually, much to my chagrin, I never took a biology class.
Really?
Yeah.
So
Goes to show you.
Well, I don't know. I think the biologist didn't want an interloper in there, and I didn't want to take introductory biology. I only wanted to take advanced biology classes. So I think it worked out fine. So
after you graduated, did you go right to graduate school?
I went right to graduate school, yes.
To do what?
Well, actually, I went to graduate school initially to do neuroscience, which was interesting but very complicated, and I really missed the clarity of physics, and so I shifted to a lab working on phage biology. which, as a historical thing is one that a lot of physicists served as their entry point to biology. Sure. And that was for me as well, even though it was, 50 years or 40 years after Delbruck did this stuff, first did it. And so I went to Rockefeller University where I worked on filamentous phage.
Was that with Zinder? Or?
Yeah, I was in the Zinder lab. I worked with Peter Modell and Marjorie Russell there.
Oh, terrific. Great people.
Wow. I just met Zinder's son.
Stephen, yeah. Did you meet Stephen, the one at Cornell? Yes.
Yeah, we were both at a ASM meeting in D.C. He looks just like his father and talks like him, too. So a PhD at Rock, and then your postdoc was at Harvard, is that right?
I was the postdoc with Rich Losick at Harvard.
Okay.
Yeah.
A nice pedigree.
Nice, very nice.
I was very fortunate, yes.
And then you came here. I remember you interviewing. And we sat right here in my office, and you knew exactly what I worked on. We had a good discussion. And, very few people, when they come for any kind of interview, whether it's a student or a postdoc or a faculty, they don't bother to find out what people are doing, unless they're in their field.
Right.
Of course, you and I weren't in the same field, but we had a good discussion about vaccines, I remember.
I remember that, too. It was very good, yeah.
So now you've been here how many years?
8.5 years.
And you're tenured, life is good?
I guess so, yeah. Hard to beat.
And so I know that you work on aspects of sporulation. I thought we'd talk about that today.
Sure.
But you didn't do sporulation in Peter Modell's, right? You did phage.
Right. I specifically looked at how phage infection turned on the sort of particular sets of gene expression in E. coli. So.
And in the Losick lab, did you work on sporulation?
I worked on sporulation. That's sort of I fell in love with sporulation as a discipline. It's great.
And that was in Bacillus subtilis.
Yes.
Which is a model organism used in many laboratories, right? And my wife did her thesis on B. subtilis. She did a phage.
Phage subtilis, yeah.
Phi 105. Yes. I could not marry anyone but a virologist. I remember in college, a friend of mine said, we have to find a good phage for Vinny. I was just thinking of that today, and we did. We found a good phage. But then she went on to parasites. And
that's right. Well, Bacillus subtilis has been a model system in microbiology since the very beginning. When Robert Koch first reported it, it was in 1877. He talked in back-to-back papers in the Zeitschrift for Pflanzenbiologie, because there was no microbiology journals in 1877. He described Bacillus anthracis, and Frederick Cohn in the accompanying paper described Bacillus subtilis. So it's been a subject of study for a very long time, and the reason that Koch was really interested in spores and why they interested in bacillus was that in the middle of the 19th century, biologists were very interested in what is life, what makes living things different than non-living things, and spores were very strange and troubling because here you had these things that seemed very dormant, that could live outside without any nutrients or anything like that. Seemed like they were not living matter, and then you drop spores into broth, and within 30 minutes you were seeing replicating bacteria. So it was a little philosophical conundrum, I think, which is sort of started the whole field going.
Are they living, spores?
Yes, probably, but very, very slowly.
Because when we have this argument about whether viruses are living, I take the position that virions, the particles, are not living, but the infected cell is living.
Right.
And people say, what about a spore or a seed? I don't know. Ask them.
Well, the spores are certainly capable of regenerating, and they regenerate quite quickly. And then there's, we tend to think of them, historically, as dormant, as not metabolically active at all. But probably a more accurate description is that they're metabolically active on timescales that are inconvenient for laboratory work.
Right.
So, enzymes, they don't grow, but they clearly, enzymatic reactions almost certainly occur, but just on timescales of days and weeks, not in the matter of minutes or seconds or something like that. And so it's very hard to actually do an experiment.
So does a spore has nucleic acid?
It has a chromosome, yeah.
Can it make messenger RNA?
Well, it's thought, the literature thinks that it can't. There's no report of it. The literature thinks that basically there's nothing in a spore. There's such low ATP levels that it can't do anything. And so it probably can't do anything on any time scale that we can experimentally measure, but there are things that go on. RNA seems to be degraded in a couple of, in over a matter of a couple of days in the spores. So, there could be that there are lots of processes going on just in sort of a slow motion kind of thing.
So what about C. diff? If I can understand Bacillus subtilis, but C. diff, which is the raging epidemic throughout healthcare facilities in the U.S.
Right.
This is really an interesting concept because if C. diff, the spore, is metabolically active under aerobic conditions, it sort of rewrites our dogma about anaerobes, if you will. Thinking about the reason C. diff is an anaerobe is simply because it can't deal with the toxicities of molecular oxygen. But if a spore is metabolically active, albeit very, very slowly, this really gives us an idea of why this damn spore is so recalcitrant in the healthcare environment and could give us, tremendous insight.
Well, I think, the spores have this fantastic external structure. They have multiple layers of protective coats, which we have some idea they exist. We don't really know how they work, but they are, the spore and Clostridium is like this, and Bacillus has these as well. They have 100 proteins that serve as these surfaces. And those probably, and that in combination with the fact the spore is largely dehydrated, probably work together to keep it so protected. The Clostridium is very interesting because, of course, it is an anaerobe, and so it can't live out on surfaces. But the spores can survive. There are reports, certainly reports in literature that spores can survive hundreds of years. There are even articles published in Nature, I'm not sure that I believe them, that claim that spores last for millions of years. They've obtained spore-forming spores from insects that were in amber. That was basically the idea of the experiment. It seemed interesting, but it's hard to actually do the careful controls in that case.
So Clostridia and Bacillus are the main spore formers.
And I think Elio thought there was you were suggesting something else? I couldn't remember.
Well, there's gram negatives that can sporulate.
Right, there's a marine bacterium. Somebody is studying it at Caltech. It's a gram-negative, belongs to what group is it? Oh, darn it. Anyhow, it's an exception. It's an outlier, so we don't have to deal with them. But most are gram-positive, all right.
Most are members of the firmicutes, which are also, things like Staph aureus and other bacteria in that are not spore formers like Listeria.
Also. Most are rods, but there's even a coccus, Sporulation, right?
Most are rods, yeah. Most are not. There are cocca ones, and there are most ones we know of are rods, but that doesn't know what's out in the environment. There are 1 million spores per gram of soil, so there are a lot of spores out there.
So for Bacillus species, the ability to make a spore is important because it lives in the soil, right?
Right.
And that helps it survive adverse conditions. But
I excuse me for interrupting. I found a paper that deals with the gram-negative spore form. It's called Acetonema longum. Mm-hmm. Belongs to the Velionelaceae, which otherwise are close to Clostridia.
Okay.
And so it's a very odd. It's an outlier, and there's a beautiful paper which we may want to discuss someday by Eliza Tosheva. Mm-hmm. Anyhow, it's great paper.
Sure, keep it in mind. So, but why
outliers? No?
Why does Clostridia have to form a spore?
Oh, why? I don't know. It's spores are a great survival strategy.
Yeah, but Clostridia's in the gut, right?
Well, Clostridia's in the gut, although it's also, actually in the soil.
Okay. All right, so Clostridia are in the dirt, so that makes sense too. But why don't all the others make spores? Why not? Why doesn't everybody make a spore? Who knows?
This sounds like a religion discussion.
Okay, so we can't answer that. Why don't you tell us how sporulation works? Or wait a minute, before you do that, I wanted to ask you, why do you study sporulation? What appeals to you about it?
I think it's pretty amazing that you have a cell that can transform itself into this dormant cell form. It has a dramatically different morphology from the normal growing cell, and that cell type can last for longer than human lifespans. And then when conditions change or improve, that spore can then exit that dormant state and start growing again. And I think that that's pretty remarkable. And that this is clearly it's not the only way to survive bad times, but it's a very, very good way to survive bad times. And the natural world is mostly bad times. So it seems this is a pretty interesting model.
So spores were what were put in the envelopes in the Bacillus anthracis thing, right? After 9/11.
Right.
Did that affect you at all? Did people call you up and ask you your opinion on?
Well, they didn't call me up at that point, but they did call up Losick and Peters, people who were working on spores at sort of a basic level, because one of the issues with spores is that they're actually very hard to get rid of. They're so resistant that normal treatment. So this is why they're a big problem in hospitals. Normal antiseptic kind of treatments, basically the spores don't care. And so actually they didn't even know how to decontaminate letters that had been contaminated with spores, because they said, well, could you stick it in a microwave? And I mean, so there was sort of a quite a bit of interest in that. And of course, there was subsequently quite a bit of funding in that area, which has sort of fallen away. But yeah.
So that's why a spore is a good weapon, especially for anthracis, right? Because it's really stable. It floats around in the air, right?
It's pretty straightforward to aerosolize, and the other thing is that you can put them in an envelope. There aren't that many biological agents that that are so robust that you could, as I say, wear on the bottom of your shoe all day, and then so that I mean there's limits with that, but they're remarkably robust, hardy species.
So how hardy are they? You could dry them out and they're fine, right?
They're fine. So we routinely take spores in the lab, and to prove that they're spores, we treat them at 80 degrees Celsius, so not quite boiling water, but pretty hot water for 20 minutes, and they have no problem surviving that.
And how long could they stay at that temperature?
I don't know exactly. I mean, it probably depends on the strain. I would imagine for at least an hour, they're probably safe at that temperature. And there are some spores of other species that actually can go up to 95 degrees Celsius, so just shy of boiling water, and survive that. So they're pretty impressive.
What's the name? Disulfo maculatum?
I think so. That may be the one that goes. There are a couple of species that seem like they're even more temperature.
Stereothermophilus is the one that they typically put in the autoclave
to test them, right?
Test kits.
Right.
So how do you make Bacillus subtilis sporulate? What do you do?
So, I mean, in the laboratory, there are two ways of doing it. One is that you grow them in a somewhat of a rich media, and then they exhaust some components of that media. We think they're probably carbon or nitrogen, and then once they exhaust that, they stop growing, and then they start initiating sporulation. And so, over the next 5 hours, 5 or 6 hours, they make a spore. You can also do it in the other ways you can do in the lab is grow them in sort of a more minimal media. And then basically shift them into a very minimal media. And then at that point of shifting them to a very minimal media, then they start to sporulate. And again, over about 5 or 6 hours, they make a spore.
I remember you saying once it's hard to get them to sporulate, or hard to get most of the population to sporulate. Is that right?
So, it's a very good question. I mean, in the laboratory, we've refined all of our methods so that they sporulate quite well. But whether or not in the real world that it's as synchronized as what we get in the lab it's probably not. And I think that's one of the challenges of microbiology, is to try to do something where you can actually do an experiment so that, for example, they all sporulate, but also make something that's physiologically relevant. And so some of the work I actually did with Michael Elowitz was trying to understand, bacteria are when Bacillus is starving, they have a number of different strategies they can do. One is to become genetically competent, which is something they share with a number of different bacteria. One is to form spores. And so how they choose between those strategies is pretty interesting and not very well understood.
A funny story, when I was a TA once upon a time in my graduate training, we had to prepare the reagents for the undergraduate lab in general microbiology. And one day we had to show the students what spores looked like in a vegetative cell. So we grew up a prep of Bacillus megaterium. And, the way graduate students do things, they inoculate a leader culture overnight and put it in the warm room and started shaking. And as I was walking it back to the lab, it obviously had stopped shaking, and the oxygen tension must have been at a critical threshold, because as I was walking back from the warm room to the lab to begin to dispense this material for the students, the entire culture lysed, and I had this beautiful preparation of spores and ghosts of where the vegetative cells came out. And so my advisor asked me, he said, what did you use to do this? I mean, we've been trying to figure out how to make spores, in synchrony and get them to auto-lyse and on cue. And you've just stumbled into something. And we never did figure out what the hell I did, but it was just one of those remarkable things that you remember from your training of the wonder. And the cell actually lyses when it makes the spore, and it releases this remarkable resting state. And we thought for a long time it was manganese that was controlling the ability to sporulate, but we tried all sorts of variations on the theme and never got that to do it again.
Can you give us now an overview of how sporulation proceeds morphologically, say?
Sure. So what happens? I mean, as most rod-shaped bacteria, as you guys well know, the rod-shaped bacteria divide in the middle, and they make two daughter cells that are identical. And the first thing that happens in sporulation is that they don't divide in the middle; they actually divide asymmetrically towards one end of the cell. So you get division results in having a large cell, which we call the mother cell, and a small cell called the forespore. And then so after that division occurs, the first thing that happens is that one of the chromosomes gets pumped in through an active process and put in that small compartment, kind of stuffed in. You can sort of think about it as there's a little machine sitting at the membrane that separates the two cells, and it pumps in the chromosome. A little bit like what happens with phages when the DNA goes into the phage head. So that's the initial thing. So that happens, and so you have a small cell with a chromosome and a big cell with a chromosome. They're identical chromosomes, or so far as we know, identical chromosomes. And then there's a very amazing thing that happens, which is that the larger cell engulfs the smaller cell. So over about a period of about an hour, the membranes of the larger cell migrate around the smaller compartment and totally enclose it. So after about an hour of this process occurring, called engulfment, you have a cell, by which I mean a chromosome of DNA, which is bound by actually two membranes at this point, and it is in another cell, in this mother cell. So it's a little bit like it's an endosymbiotic kind of process, right? And so you have this forespore now totally contained within the larger mother cell, and then the mother cell is responsible for synthesizing all of the surface structures of the spore. So it kind of assembles that over the next hour or so, and there's, as I said, about 100 different proteins that are added to the surface. And then once that's complete, the mother cell autolyses and dissolves away, so leaving just the spore. Whether that's a directed process of sort of suicide, or whether just because the mother cell exhausts itself and sort of basically lyses in a passive fashion, we don't really understand. But if you do in the cell, most mother cells lyse without having to do anything to the culture.
So do we understand how the reduced nutrients are sensed that starts this whole process?
So it's a really good question, and we're somewhat embarrassed to know that we don't understand very much what the proximal stimuli are. We know that limiting conditions of carbon and nitrogen sources probably contribute to this. Mm-hmm. There have been various reports that a fall in GTP, guanosine triphosphate levels in the cell are key, but those experiments are suggestive, but certainly not conclusive. So we don't actually.
There's an interesting protein, isn't it, called Y?
Yes.
That's
which senses those levels, and that plays an important role. But there are other things, and the way the cell senses this actually is there's what's called the phosphorelay, which is actually similar to what you see in eukaryotic cells, where there's a kinase that sits on the membrane typically, and that senses something, although we don't know what. And then when that kinase gets active, it proceeds through a series of proteins to activate a transcription factor, which then gets sporulation going. There's a master regulator of sporulation, which is the DNA binding protein.
So that's SPO 0.
SPO 0A, right.
SPO 0A. SPO standing for sporulation.
Right, and then 0 as in, and so these were originally, the original genetics of sporulation were done in the 60s and 70s, where they classified mutants based on what stage of development they were blocked at. So there's the SPO 0 mutants, which are blocked at the first stage, even before this asymmetric division I told you about. And then there's SPO 1, 3, SPO 4, SPO 5, SPO 6. And so there's a whole set of mutants in each of those stages.
And these, but the first one, the SPO 0A, is a transcription factor.
Yes.
And this turns on a lot of other genes that are all participating in the formation of the spore.
So it turns on about 100 genes. It has a regulon that's estimated to be about 100 genes, which are sort of get things rolling. And then there's what was identified starting in the 70s, that there's a series of transcription factors that are in each of these individual cells. So this is why sporulation is also a wonderful system. It's sort of a model developmental system, because you have these two cells which have the same identical chromosomes, but there are patterns of gene activation that are completely different and specific in each of those compartments.
Okay. So you said that a chromosome is pumped from the mother cell into the spore through a portal of some kind, right? So does cell division, DNA replication occurs just before the spore formation?
It's not. It's a really good question. We think that actually replication is already completed.
Okay.
And that there's there's a lag time between actually completion of replication and when the chromosome was pumped in. But being certain that the chromosome was 99% complete versus 100% complete, it's hard to differentiate those.
And what else goes into the spore besides the chromosome? Anything else that we know of?
We don't think that there's much that's going in. Most of the DNA binding proteins that are on the chromosome seem like they're getting stripped off the DNA as it gets transported. There's this pore that is a ATP-driven machine it's a multimeric homomultimer of a protein called SPO3, which actually has homologs in the way plasmids get brought into bacterial cells. Mm-hmm. So this is a well-conserved process. It seems like the pore would only allow a double-stranded DNA through.
Right.
So it seems like specifically things aren't included. Now I should say though that there is a pore at this stage. There actually is a pore at a later stage in sporulation, which we think, although the evidence is still coming in, that there's a pore between the two cells. That the mother cell not only produces or is responsible for making proteins that coat this and structure the spore, but actually provides nutrients to keep that spore alive until, in a true nursing fashion.
Mm-hmm. Mm-hmm.
And so in addition to moving the chromosome into the spore, because anthrax is virulent because of a plasmid,
right?
They also pump plasmids in, too.
That's a really good question. They must.
They have to, because otherwise an anthrax spore wouldn't be virulent.
Right, and they're totally virulent. So I don't actually. It's a really interesting question. How and I should know this, but how the anthrax plasmids, of which there are two plasmids that are important for virulence, are appropriately transported. Because in Subtilis, of course, we don't have these plasmids, and we don't actually have any plasmids. But actually, most spore formers have multiple plasmids. For example, Megatherium, I think, has 9 plasmids in natural form. So, it's an interesting question.
Because if you think about it, it could really, change the virulence profile, because if you don't get the right partition of the right complement, if in the case of anthrax, you need the 2, but in Megatherium, in order to confer the full genetic profile of Megatherium, if it doesn't partition all 9 of those plasmids properly, a fraction of the population of spores could be, if you will, for lack of a better term, avirulent or aphenotypic.
Absolutely. No, it's a very good point. And in fact, I may be speaking out of total ignorance. I'm not aware of this, but there could be, people have investigated this, and I just don't know. It's a good question, though.
So in this process of sporulation that you described, at what point, if you add nutrients, does it not reverse anymore?
Oh, yeah. I'm laughing because this was actually something I worked on as a postdoc, actually, in Rich's lab. So it's a subject that's pretty dear to my heart. So when they've made this polar septum, they will actually, now if you take those cells and you give them rich media, like Luria broth, they will actually stop sporulation and start growing again. You get these actually fantastic looking and sort of weird looking cells where it's almost like there's heads growing out of this small compartment because they've actually started to grow again, which they don't normally do. Once they've started the turning on genes, specifically in each of these compartments, in the forespore and the mother cell, then it doesn't matter what you do. It's a developmental commitment. So once they've really differentiated, by which I guess you mean that the genes are differentially expressed, that's it. Doesn't matter what you do, they are going to complete sporulation, come hell or high water. So
you'll get the spore coming out.
Right.
But in general, the from a population point of view, the story is a little bit complicated because sporulation is a great way to resist harsh chemicals and high temperature and so forth, but it's a lousy way to take advantage of good times. The spore is, unless it germinates, isn't going to participate in the game. And so the bugs don't take advantage of a good situation necessarily. So commitment to sporulation has to be done in a thoughtful way. And I guess Outliers does just that. It weighs the possibilities, and not everybody sporulates, and it's a complicated story, isn't it, Jonathan?
Well, I think is that you're exactly right, Elio. I mean, I think what happens is that they basically go some distance and they have some flexibility. But as I'm describing these morphological changes, the cells are very different, and it's a little bit like if you build a house, right? I mean, if you stop halfway through and you haven't put the roof on, it doesn't do you much good to build the foundation, right? And so I think, basically at some point, they say, look, like, I know this may not be the right thing to do, but I got to finish it because otherwise, you're dead, right? I mean, it won't do you any good. So there is sort of a point of commitment that I think is really, really important for the strategy. But, that basically this is a long process. I mean, this is from the lifespan of a bacteria that can actually divide every 15 minutes. This process of sporulation is 5 or 6 hours. So they really and when this point of commitment comes pretty early in this process, it comes at approximately 2 hours into this process. So they still have 2.5 to 3 hours where they can't do anything except finish building the structure. So if conditions change at that point, they're pretty much they're vulnerable.
And in the case of the Clostridia, that had perfringens has a generation time of 9 minutes.
Yeah,
it really drives home the.
Does?
Yeah, 9
sure
minutes.
That? Hey, wait a minute.
9 minutes.
Wait a minute.
Perfringens has a generation time of 9 minutes. It cheats. It has multiple copies of everything.
That sounds like Elio doesn't believe you, man.
I think he doesn't.
There's a Vibrio nitrogens, which in the literature that I know of is supposed to be the world champion fastest grower, and his generation time is, I think, 13 minutes. So this is news to me. You're probably entirely right. I don't doubt it for a minute, Michael, but I didn't know this, and that's so. All some
right, I just asked Dr. Google, and I went off to Health Protection Agency of the UK, which came up first. Under optimal growth conditions, the organism has a generation time of 10 to 12 minutes.
All right, that's close enough to mine.
That's C perfringens. So my neurons are functioning again.
Sure are. They sure are. Well, thanks for enlightening us.
So you mentioned that at the end of the whole process, the spore, which is developed inside at one end of the bacillus, then ruptures the host, right? Do we know how that happens? There must be some enzymes that digest away.
There seem to be enzymes that digest the cell wall of this mother cell of the larger cell, and it's basically like an antibiotic, right? I mean, it's the same thing, the same kind of mechanism that penicillin would do, and that seems to be the process. But there is some question as to whether or not this is really an active process, or it could be it's a cell that essentially gives up the ghost, and there are holes that happen in the cell wall just as by normal sort of things, and then they just fade away. It seems unlikely, though, that I'm not a firm believer that things just happen by chance. I mean, I think there's a directed, mechanism. So
okay. So what aspect of sporulation are you focused on? You mentioned it earlier. Tell us again.
So I'm really interested in the whole. So we're interested in dormancy. We're interested in what the cell does to begin to prepare itself to become dormant, the spore, and then we're also very interested in what causes the dormant spores to germinate, to start to grow again. I think this is a really fascinating issue that not much is known about in microbes, because of course this is such an important decision, because if they decide too early to start growing again, then they can be vulnerable and can be killed. But of course, if they're too conservative with that decision, then their neighbors are going to eat up all the nutrients and take over the population. So it's clearly a very important decision to be made.
And how is that made?
So in one of the things that my lab is worked, so I'll talk about the germination issue, and we've sort of, people for a long time, starting with Koch and Kohn in the 19th century, people have considered the role of nutrients as being detected by spores to cause them to germinate. And in the laboratory, for sure, you can get spores to germinate with high levels of certain amino acids like alanine, sort of millimolar levels of alanine. It's not clear where in the environment or in a host that you would see millimolar levels of an amino acid, but those work very well. They germinate 100% of the spores. But
they're called germinants, aren't they?
They are called germinants because they induce germination, although it's not exactly clear what the mechanism is. But we took a somewhat different perspective, which said, well, why would spores ever want to germinate? And nutrients are one possibility, but maybe they're also detecting other bacteria in the environment, and specifically that there are other bacteria that are growing. And so then begs the question, well, how can they detect that? And it's actually a little known, but I think a very interesting observation, that both gram-positives and gram-negatives, to a lesser extent, release substantial quantities of cell wall material as they grow. It's a little bit like molting that snakes would do, right? As they grow, they can't totally fix their peptidoglycan and conserve it, and so some of the peptidoglycan gets released into the milieu, and this happens during growth. It's a natural consequence of this assembly. And so we said, well, maybe spores detect growth of other bacteria in the environment by this peptidoglycan. And so we actually found, in fact, that spores do detect peptidoglycan. They detect chemically synthesized parts of peptidoglycan or peptidoglycan that we purify from growing cells. And so we think that that's an important signal. So it's sort of a quorum sensing signal in the sense that they're detecting bacteria in the environment, but they're most importantly detecting that they're active and growing.
So if you add peptidoglycan, that's enough to get them out of dormancy.
That will be sufficient to get them to exit dormancy.
I should say this was an amazing phenomenon, and it was your discovery, Jonathan.
Well, thank you, but yes.
Really a beautiful piece of work.
So have you ever thought of using ground up peptidoglycan as a spray to get C. diff to germinate in the hospital to die?
I haven't actually thought about it with C. diff, but I have actually had been contacted by members of the U.S. government, shall we say, who are interested, because of course that is the problem with things like anthracis, because no one knows how to get rid of spores, and if you can germinate things like anthracis, they're just as vulnerable. They're vulnerable to all sorts of things. You can wipe them off with a wet tissue paper. But until you do that. So actually, there is some interest in figuring out ways to do that. The peptidoglycan is a very interesting molecule, and it's actually fairly labile in ways that we don't totally understand, to be honest. So it's not a good thing, but if you could figure out a mimetic that was chemically stable, that would still stimulate, I can tell you some people who would be very interested in that.
Yeah, I'm sure. So
you're something of an expert on chemical modifications of peptidoglycan, aren't you?
Well, not an expert, but we have worked on it. There are a lot of very interesting modifications, yes.
What senses the peptidoglycan in the spore, right?
So that's really interesting. So it turns out that what senses the peptidoglycan in the spore is a membrane kinase, which has as its extracellular domain something called apostata repeats, which are seen in some other bacterial proteins. These bind peptidoglycan, and the intercellular domain of this protein is a kinase. It's a very interesting kinase, and that's actually been the subject of a lot of work in my lab in the last couple of years. It's a serine threonine kinase that looks just like eukaryotic serine threonine kinases. So bacteria have signal transduction pathways. They have serine threonine kinases and serine threonine phosphatases that look structurally basically identical to eukaryotic kinases and phosphatases.
But here's the big question.
Yeah.
How can this poor kinase sense the outside when it's surrounded by cortex and this and that, all kinds of layers around it.
Well,
how can you go out that far?
It's a great question. And actually what turns out is that I think, well, there's two things. One is that the neuropeptides that we think it senses, that it can certainly bind to in vitro, are small enough that they probably penetrate this. So, in the textbooks, the spores are written as sort of having this impenetrable coat. But actually, it turns out that if you actually look at the literature, molecules smaller than 5 kilodaltons can probably cross these layers. So it's very good for keeping out protein. So that's actually why spores are resistant to lysozyme, because the lysozyme can't gain access to the peptidoglycan that protects them, because it's held off by this coat. And the other thing is that, which we don't totally understand, but I think is very exciting, is that the spore actually is not a static structure, and it seems to be a somewhat dynamic structure. It almost looks as if it's sort of breathing in and out. This is difficult to visualize, you can imagine, because these spores are, a micron in size, and so our methodologies for seeing this are really cutting edge. But I think there is some good evidence that there's actually dynamics in the structure.
So when this kinase recognizes peptidoglycan, it starts a phosphorylation cascade?
It starts a phosphorylation cascade, which to be honest, we thought at one point we understood. We think it directly regulates translation in the bacteria. And we have some evidence that it regulates the activity of elongation factor TU. Spores, like many dormant cells, have actually pretty large storehouses of RNA. That's interestingly not very limiting, or at least doesn't very limiting. And so we think that the spores are set with, basically they have RNA and they need to start translation. And so we think that this signal that comes on through peptidoglycan starts translation of the RNAs in the spores, and that's the beginning of that's where the whole thing starts. We're not quite there in establishing the whole signal transduction cascade, but it seems like it's going to work out something like that.
So they're translationally dormant.
Right.
And then you turn on somehow EFTU.
Right.
And that gets them going back into vegetative phase.
So we've had a lot more success looking at, so it's hard to work with spores because, well, they're designed not to be messed with, right? That's what their whole raison d'etre is, to be robust. And so actually that makes them not great things to use for biochemical assays. They're not easy to break open. So we've been actually studying a lot with how the converse question, which is how do spores become dormant? What causes them to become dormant? And we've had more success with that because those cells are actually much more tractable. Until they reach this final stage of dormancy, they're actually more like regular spores. And what happens, and this is work that we haven't published, but we're very excited about, is that actually during sporulation, the spore turns off translation. And it does that actually by regulating the phosphorylation state of this protein EFTU. And so when EFTU gets phosphorylated, it gets inactive. And so the process, if you look in spores, they actually become translationally inactive as they proceed through sporulation. And we think that eventually comes to a stop when they're finished. At some point, they just completely stop translation altogether.
So remind us, what does EFTU do in translation?
So EFTU is a key protein. It's actually the most abundant protein in bacteria. So it's probably one of the most abundant proteins on the planet. And it is responsible for delivering the tRNA to the ribosome. So it performs an essential function. And it's every time there's a new codon, an EFTU has to be engaged.
Okay. So can you delete the gene for this kinase, the sensor kinase? And are those bacteria viable? And then will they go and make a spore but not be able to be resuscitated from the spore?
So it's a great question. So we can delete the kinase. And what happens is that actually it seems to be, at least in the laboratory, that the spores are more or less okay. But they are no longer responsive to peptidoglycan as a germinant, but they still respond to alanine. So it seems like, which is maybe not surprising, that there are, this is an important enough decision that the cells probably have a number of signal transduction cascades, and there's a number of signals that are going on.
So it's not just peptidoglycan. That's great. That makes perfect sense. But the vegetative bacteria doesn't need this kinase to survive, right?
It doesn't need this kind of stuff, but, I should say that this is, maybe something that touches on other things that you guys have talked about in this series. But, we study bacteria in a very strange way, right? We study mostly what's going on in log phase, in laboratory conditions, 37 degrees, all the nutrients. And it's clear that most microbes don't live that kind of life. They are living they're very nutrient limited in most conditions. They sort of manage to squeak by. So under laboratory conditions, this kinase is not essential. But what's interesting is that if you delete the kinase, the bacteria grow fine in log phase, but they die in stationary phase. Mm-hmm. So it's this question. I mean, I don't want to get all metaphysical, but the question of what's essential, right? I mean, we think about, it's in log phase, then it's not essential. But stationary phase is what most microbes exist in, right? So if things die in stationary phase, then that's a pretty essential, I think, a phenotype. So,
is this kinase present in all bacteria or just those that form spores?
So the particular form of this kinase, which binds peptidoglycan, is present in all gram-positive bacteria. There are potential homologs and gram-negatives, but they're sufficiently different in how they bind peptidoglycan that we don't know. But there are kinases like this in all bacteria that have been examined. So we think we're hopeful that this is a general mechanism by which bacteria can slow down their growth and maybe in cases become essentially quiescent and then in subsequent times start growing again.
So do you think that this kinase system, if you think about the holy grail of microbes that are always in maximum stationary phase, even when they're in exponential growth, which I always view as mycobacteria, do you think that if you can noodle on this sensor kinase regulon or the associated proteins, we may have a new target for an antibiotic for TB?
Well, I mean, it's a great question. In fact, one of the things we got very excited about early on, it's been slowed because we're not experts in TB, and as you know, it's a very hard organism to work on.
Oh, yeah.
But one of the key observations with TB is that there are proteins called resuscitation promoting factors in TB. You may have heard about them. These are proteins that are essential for sort of viability of TB in various host models. These proteins are very interesting because it wasn't known what they were until they were crystallized, and they were seen to be essentially lytic transglycosylases, in other words, proteins that could digest peptidoglycan. So one of the things that we were very excited about was whether or not the RPFs, these resuscitation promoting factors, were working to cause MTB to start growing again by digesting peptidoglycan and making the signal that would then go back and start them growing again. It's doesn't seem like it's maybe that simple, but it's a very hard organism to work with. It takes 3 weeks to get a colony on plates, which is, for someone as impatient as I am, a frustratingly long time. So,
by the way, some years ago, somebody came up with the tantalizing idea that mycobacteria make spores, and it was your ex-mentor, Rich Losick, that didn't like the idea. Is there any new developments on that, or is it?
No, I mean, I think the evidence is the evidence was not great to begin with. It was certainly an alluring possibility. But, I mean, what's interesting is that I think the evidence they had was some pictures, which are always sort of interesting, but it's hard to know how to evaluate them. And they also identified a bunch of genes that they thought were homologs in MTB to the sporulation specific genes in Bacillus subtilis. But it turns out that most of the genes they saw as homologs had homologs in other bacteria as well and probably were doing different kinds of functions. So I think it's pretty accepted that MTB is not making spores. On the other hand, I should say that even though they're not making spores, there is pretty good evidence that they can exist as dormant species for a long time. One thing that's classic with TB is that people can have these, granulomas where there is not a lot of bacterial growth for their lifetime. And, they can basically get infected when they're a child. And these things don't reactivate for 50 or 60 years. So, it may not be, they're not spores, but they're a pretty impressive dormant or quiescent species.
They resist drying. I mean, they can be found in dust. If somebody coughs up TB, they stay in the room for a long time because they don't die on drying.
Right, they don't dry. And that actually is the case. I mean, it's a great point, Elio. I mean, actually, a lot of bacteria, it turns out, have states where they actually are pretty, they're desiccation resistant. Legionella does this. I know there are a number of bacteria people have looked at where you actually, if you treat them if you do a proper drying them down and stuff like that, they actually will resuscitate quite well. So this seems like it's a pretty, maybe more general phenomena of microbes than we've sort of hitherto appreciated.
So this resuscitation promoting factor is really, it may be a universal function that bacteria have to get them to move out of stationary phase back into a logarithmic phase of growth.
Right. And actually, that is those kinds of proteins, which, MTB, they're called RPFs, but, Bacillus subtilis and all other gram-positives have homologs those. And in fact, Bacillus subtilis has one of them that we've studied in the lab, and that autoregulates itself via this kinase that I've been telling you about that is responsible for germination. So you asked what this kinase is doing in log phase. It's not essential, but it actually serves as a signal transduction pathway where it induces the expression of this hydrolase, this thing that destroys peptidoglycan, in response to there being free peptidoglycan.
I wanted to just ask you a few questions about another paper that you've published. Before we do that, any other questions before I move on?
I think this was a wonderful explanation. I'm really so delighted to have heard this.
Oh, thank you, Elio.
Thank you, Jonathan.
You have a paper in collaboration with Michael Elowitz's lab called Pulsed Feedback Defers Cellular Differentiation. And I just wonder if you can explain what the general idea here is that when you nutrient starve a Bacillus subtilis, it takes a while before it sporulates, right? So what's going on?
Well, I think we're the sort of textbook model of what's going on is that that the cells basically decide all in one cell cycle to start this developmental pathway,
right?
And there's this transcription factor that I'm telling you about called SPO08, which is the master regulator for kicking off sporulation. And so the idea was that basically that thing got phosphorylated, and then that was it. Once, and then that was sort of the key thing. And then I think in part with this paper, and there's observations from other labs as well that have come out in the last couple of years, it's suggesting it's not that simple. And the idea is that actually, if you look and some of the experiments that are in this paper, and as I said from other labs, show that actually this phosphorylation actually occurs much before this starvation event occurs. That actually the cells are, maybe you can say, sort of sampling the environment, and they're going through cycles where they go up, they increase phosphorylation, then they decrease phosphorylation, they go up, and so one argument for that is that there isn't such a dramatic thing as like this switch gets turned on or turned off. It gets turned on a little bit, and then maybe if things get really bad, it goes up more, and then if things get a little better, then it goes down. And I think this idea of the sort of black and white transitions between growth and something like sporulation or something like competence, another response to starvation conditions, is I think it's clear that's not the simple model is going on. That there is kind of if you look earlier on, and I think now that we have these very nice microscopy techniques that Michael and his colleagues have pioneered, where you can actually follow single cells, you can see, in fact, that these transcription factors are getting turned on or getting phosphorylated in cells that are like 5 cycles away from ever becoming a spore. So these things are happening, but you wouldn't see that because, of course, when we're thinking about sporulation, I told you in the earlier part of our discussion, we only can see this because they actually divide asymmetrically. But that's an hour and a half into sporulation. They've committed themselves to going, or more or less committed themselves to going at that point. But what we're seeing now is we can look at individual cells, and I think this is a very exciting part of microbiology. You can see that there are things going on in these cells where there's no obvious, gross morphological changes that you can observe, but there are changes in the levels of these transcription factors that are indicative that there actually is a lot going on. They're not simply doing it all at once.
And you can look at individual cells because you hook up fluorescent reporters to promoters that are controlled by certain transcription factors. Right. Is that how it works?
Yeah, so you can look at and so that that gives you a key. Before we had this these kinds of approaches, you could look at a population of cells and say they were doing this, but and that there are very nice things about a population, but it's really an average, right? And so if not all cells are doing that, it's not easy to tell that. But with the single cell thing, I view it as it's making microbiology into an organismal biology science as well, because we're really, we're looking at what's going on at this cell versus another cell, and they're in the same, these are not different bacteria. They've actually been growing under the same conditions. They're on the same microscope slide, and what's really striking is that they do very different things. And I don't think that was, I don't know. It seems to me it's so obvious that we didn't think that was going on that I couldn't even imagine until you actually see these bacteria that are responding very differently to what you think of as the same, same conditions.
Yeah, the figures in this paper are beautiful with these red and green fluorescent proteins. I particularly like this picture, a time course from 0 to 24 hours, and the bacteria start out black, and then they turn green, and then you can see the spores forming beautifully. And they're all different shades of green.
I think that was, when it was about 10 years ago that Michael and I started making movies of subtleist sporulating. And I think one of the first things that we were struck by was how heterogeneous the population was. We're, as microbiologists in these bulk cultures, we're used to thinking about, they're all doing this at 2 hours after some treatment, they're all doing this. But in fact, they're not. And I think what's really interesting is not just that they aren't, but I think the biological implications of that there really is heterogeneity, which is good because, if they may not, some of them may not make an accurate assessment, and some of them may be very aggressive in making the assessment. But in the end, what you want is some of them to survive, right? That's the goal of the population. So this sort of fits in with this thinking about them as real cells, as individuals.
Yep. I think the same happens in other systems, say virus infections as well. We study populations of virus-infected cells, and we never look or rarely look at individual cells. But if we did, we would probably find some very different stories.
Right.
But we don't do that. It's probably worth doing. Anything else, gentlemen? Are you all satisfied?
I tell you, the first time I heard about sporulation back in the middle 80s was from Rich Losick, and the cascading sigma factor story impressed me and showed me the elegance of bacterial gene expression, and the subsequent 25, almost 30 years now, watching it evolve into this elegant system, it even further reinforces how truly amazing bacteria are.
They are. That's why we're all here, right?
Yeah. Without them, we'd, have a dull life.
Okay. I want to read 2 emails here before we finish up. We get a lot of email from listeners, and they're wonderfully engaged. The first one is from Jeff, who says, "Hello, TWiMers. In TWiM 26, I heard the listener email asking if you would consider offering continuing education credit for your podcasts. And you understandably cited the work that you would need to do such, as writing learning objectives, tests, etc., as a real barrier. I was wondering if you would consider contracting that kind of work out to some of your listeners. Being a teacher of microbiology class at a community college, I spend a large amount of time coming up with learning objectives, tests, etc. It would help me to generate fresh material for my students from your podcast. I wouldn't be surprised if there were others in your listening base who might feel the same. This still would add work for you, so I understand if it's a no-go." Yeah, well, if anyone wants to volunteer, we'd be happy to have them do it. In a separate vein, quite a while ago, I was doing dishes and listening to TWiM. As I was using a sponge to wipe off the counter, a ridiculous question popped into my head, as opposed to my toe, I guess. Are there strains of bacteria that can provide a protective commensal role on environmental surfaces? A countertop is nothing like a biological surface, as only the latter should have organic material to eat on it, but still, is there any evidence in any situation that something like this happens? Please read the following sentence with extra emphasis. I am not fishing for an excuse to get out of doing the dishes, and I regularly boil our kitchen sponges and use bleach to clean the counters. Keep up the great work.
The short answer is skin.
There is a study done by somebody in Israel using phages. Now, we had this on Small Things Considered, and I don't remember the story.
Yeah.
But there is, there's a use of phages that will influence, can be used on surfaces, and it was done that way. I'll be looking for it.
You're talking about
I'll look for it.
You're talking about cleaning surfaces with phages, right?
No, it's more complicated than that.
Yeah.
I'm fuzzy, and let me get off it, but I'll look for it.
Okay. Michael, you said skin?
I was going to say skin simply because, every human, Staph aureus is thought to be our Staphylococcus. Specifically, the coag-negative variant is a microbe that we have as a component of our skin flora, and it's obviously occupying a particular niche. Similarly, if you look at other mammals, they too have other staphylococci that are not Staph aureus, the coag-negative variant. There are other species of staphylococci. So surfaces, at least human surfaces, do indeed have a normal flora, as opposed to inanimate objects. You could almost make the argument that if you have a benign organism on the surface, and your likelihood of picking up a benign microbe as opposed to a virulent one would be serve you better, because the likelihood of you encountering the man-eating bacterium would be less, because you're simply running into the the nice little pet that happens to be on the surface.
Oh, your granite countertop probably doesn't. Well, it's probably got something on it from all the food you cut on it, right? But
yeah, the granite countertop does, but it also emits radiation, so that helps to sterilize it as well. Well, the incident radiation coming out can,
yeah, sure.
It's a rock after all, and there's, God knows what's in rocks.
Bacteria, probably.
Yeah, that's geology.
Aren't there bacteria that grow in rocks?
There are spores that are been recovered from gold mines in South Africa. So
really,
yeah.
So can you use these ancient spores to see what genomes were like, a million years ago?
Yeah.
If you believe that they can live that long, I'll just close in my discussion of spores with my favorite example of environmental spores. So it turns out that when it was not Amundsen, but Scott did the expedition to Antarctica, and as a good Scotsman, he brought with, of course, all the essentials for it, including scotch.
Yep.
And there was in one of their huts, which of course they never got to because they died, there was like 10 cases of scotch. And they, of course, are wooden cases of scotch. And 100 years later, they found one of these sheds, and they found these cases of 100-year-old scotch. And they found spores in the wood that were spores from Scotland.
Wow.
So the spores had survived the trip and had lived there for 100 years. So I think that's that to me is the best example.
That's a good one. Yeah. It's not a million years, though.
But it's scotch, so it's good.
The other email is from Velma. Dear TWiM team, greetings from a longtime listener and fan of all three podcasts. I'm a biotechnology master's student at the University of Helsinki, Finland, but I earn my living as a part-time driver in the world's most northern metro. Wow. A subway driver? Is that a metro, right?
Yeah.
Cool. Your podcast helped me pass the dull hours at work so that I feel like I'm doing something to advance my studies as well. Well, pay attention to driving, I hope. I've been thinking of writing for quite a while. Now that I'm finally doing it, I have a rather general suggestion and a link. The suggestion is I'd like to hear more about the archaea, maybe even an entire episode dedicated to them. They seem like such a mystery. At least at my home university, they're almost always just mentioned as an aside, while the bacteria get all the attention. The one time they were actually discussed on a course, I learned that they are not just extremophiles, but actually all around us, just like bacteria. This made the fact that there are no pathogenic archaea seem even more curious to me. Maybe we just haven't found them yet, right?
Well, there's thoughts that some of the oral microbes, which are archaea, do indeed cause periodontal disease. So it's
But
just
that's about it. It's kind of it's very surprising because it's one of the great mysteries. I mean, this is a large group of organisms that really, in many ways, resemble bacteria, at least in shape and form. They share some habitats. Some of them are host adapted, and why don't they? I mean, it's not. Somebody once wrote, who was it? Ohio State wrote an article. John Reeves wrote a whole article on it, and he asked, why are archaea not pathogens? And at the end of it, he spent about 3/4 of the article talking about it. And at the end, he said, I don't know. So, good questions. Zelma, it's a great question. We don't know.
So would we like to have an episode on archaea sometime?
Yeah, I think it would
so
be
many
a
aspects to it.
Many aspects,
fascinating
course,
topic.
Yeah.
We could talk about methanogens. We could
Yeah.
talk about the halophiles. I mean, there's
Hyperthermophiles.
Yeah. All right. We'll get some guests who can talk about that. As for the link, it's for the Earth Microbiome Project. I just ran across it today and don't remember this project being mentioned on TWiM before. I think the idea is awesome, not to mention really ambitious, maybe even overly so. What do you think? Is creating even a rough map of the microbiome of our entire planet an attainable goal? And would it be a useful resource for research? Thanks for the inspiring and informative podcasts. What do you think about this? A systematic attempt to characterize the global microbial taxonomic and functional diversity.
I think it's too difficult. I think there's just too much. Just like, I mean, there are who knows how many species, so-called, of bacteria, but it's probably in the millions, if you consider species in a broad sense at least.
Yeah.
And it becomes a huge investment in data gathering. And I don't know. It seems to me that it should be a bit more targeted.
What do you think, Michael?
I think that it's an interesting question, but it'll just be a catalog. I would much rather look at the relationships to the microbe and the environment, whether it be a living organism or whether it be the environment itself, to understand how the microbes are shaping our planet and our lives.
They're going to analyze 200,000 samples from various communities around the globe and make 500,000 reconstructed microbial genomes. Well, if they get
that, by the way, is that's a bit elusive. So far, the reconstruction of whole genomes from environmental samples has been pretty limited to acid mine wash, and some efforts are being made for hypersaline environments. But for the rest of it, it's not an attainable goal yet. It's just
why is that? It's technically hard.
Well, that's right. I mean, you have too many sequences.
Yeah.
In order to link them together, you have to have a huge, powerful amount of information.
Yeah.
And it's just not there. So
it's not just 16S RNA sequence; it's the whole genome they want to do.
So they're talking. She's talking about if you do the giant metagenomics of the meta-Earth, you are going to end up with metadata. You know, they're just factoids which are going to clog up a computer. I don't think this is I don't think it's a way to go yet. If and when it becomes possible to assemble genomes from metagenomic data, that is from bits of DNA, in a sensible way, then I think it would make sense. But right now, it's just a gigantic plate of broken up spaghetti.
Well, I love spaghetti. You do too, I bet.
Sure.
But maybe this will force the methodology to get developed. You know, you never know.
But it's being worked on. I'm
sure.
This is an active area of work.
All right. Thank you, Velma. You'll be able to find this and all the other episodes of TWiM at iTunes and at microbeworld.org/TWiM. If you do like TWiM and you use iTunes, go over and leave a rating there. That helps to keep us on the front page of the iTunes podcast directory so more people can find us and learn about the wonderful microbial world. We'd love to get your questions and comments. Send them to twim@twiv.tv. Jonathan Dworkin is here at Columbia University. Thanks for joining us, Jonathan.
Thank you, guys. Thanks.
Hope you enjoyed it.
I did. I had a great time.
Our listeners will love it.
I hope so.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure as always. It's always fun.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks. And it was a great time yet again.
Yep. I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, Chris Kandian, and Ray Ortega for their behind-the-scenes help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.